A very good day everyone. Today in this video we will be discussing about some of the important landmark experiments which have taken place in developmental biology. This section comes under your unit number 5 in CSIR DBT NET examination. So why are these kind of experiments considered important? First is that these experiments they helped in establishing some of the very basic fundamental principles of the entire developmental biology. The second is such kind of experiments helped us to explain the cellular fate. What is cell fate? What do you mean by determination? What is embryonic induction? So these kind of important terms, important concepts came into picture once we did these kind of experiment. Third, it helps us to introduce some very important concept like organisms. How are the cell regulating themselves? What are morphogen? What are cytoplasmic determinant? How are the genes getting regulated in order to form an entire embryo from a single cell stage? And another point is it helped these kind of experiment they have helped us to form some of the very basis of the modern stem cell theory which we all study, and also it gave us a very in-depth understanding about the regenerative biology. So let's have a look at all the different kind of experimentation which has been done in developmental biology. The first set of experimentation is what we call as the nature of cell fate. That means in these kind of study, these kind of experimentation, we will get to know what is the meaning of a mosaic development, what is the meaning of a regulative development, whether a embryo is pre-programmed, that means whether it is preformed, or what do you understand by point mosaic development, or does an embryo have an adaptable regulative development. So these some of the very important experiment of our very initial stages when the techniques were not so high class. So they tried their best to give us some of the very basic landmark experimentation in the entire developmental biology. The first is Wilhelm Roch's mosaic development theory, which he gave in the year 1888. So in this experiment, what he did is he took a two-cell stage frog embryo or blastomeres of two-cell stage frog embryo, and he destroyed one of them by using a hot needle. He took a hot needle and he punctured one of the blastomere, and then he was observing the development. The remaining cell, this other half of the cell, he found that it developed into an embryo, but it formed only half the embryo. So the observation what he made was that he could see the formation of only partial embryo and not a complete full embryo. The conclusion which he proposed was he first gave the term of mosaic development. He said that each cell, he had two blastomeres, so he said each cell contained some information which was predetermined, and hence each cell contains predetermined developmental information. So if you destroy one of the blastomere, so that particular 50% of the information was lost, and hence you have only half the embryo. He said that embryos they have autonomous specification development. That means the cell fates are strictly divided during the cleavage period. And since you did some kind of experiment during the cleavage period, you destroyed those kind of information, maybe. And thus the cell they act independently of their neighbor. So this cell it died, this cell survived, and hence it could give rise to a half embryo, the information which was present only with itself. So the significance of such kind of study of his experiment specially is considered as one of the first breakthrough experimental approach in developmental biology. His theory was later challenged by another scientist known as Driesch. So the next experiment is of Driesch only, known as Hans Driesch sea urchin experiment, which he did in the year 1892. So the experiment what he did was he took again two blastomere stage of a sea urchin embryo this time, and he observed that each cell developed into a complete larva if he separated them. So the conclusion what he gave was somewhat contradictory to the previous one. He proved the term known as conditional specification or regulative development. That means the cells, they are in a tendency to communicate with each other, and thus they can change their fate. It depends upon which kind of experiment they are placed in and what all are the neighboring cells around them. So his experiment is considered as important because he gave the term developmental plasticity. That means the cell, they can change their cell fate during initial stages. So thus the term of totipotency came into picture. If you have a, suppose you have a 4-cell embryo, 4-stage blastomere, and he separated them. Okay, if you have 2, you separate 2. If you have 4, you separate 4. So he found that exactly 4 normal uterus larva was formed from the sea urchin experiment. So he said that there is some kind of conditional specification or regulative development. They could individually regulate their growth so as to give a complete larva, unlike the previous one. The next important experiment is Speamann's hair loop constriction experiment, which was done in the year 1901. So what he did was he also took the embryo of a salamander egg, and he used a baby's hair, a fine hair, in order to make a constriction. So he observed that if he made constriction and the two portion, they received nucleus, if nuclei entered into both the segments, so both of them develop into a complete embryo. That means he was the one who gave a concept saying that nuclei is the one which carries all the hereditary information which is required for the development of an embryo. And this formed the very basic foundation of nuclear equivalence theory. That means the nucleus contains all the hereditary information, all the necessary information for which is required to form a complete larva. Now in this also he did various kind of case experimental study. For example, we know there is a region known as gray crescent where the male gamete comes and fuses with the ovum. So the ovum shifts a little. The cytoplasm turns a little, approximately 30 to 35 or 45 degrees. And this leads to the formation of a gray crescent and distribution, unequal distribution of cytoplasmic determinant. So in first case, what he did was he bisection along the gray crescent plane. So in this gray crescent itself, if you do a constriction, so he found that even then two normal embryos or two normal larvae could be formed. That means both the cells, they had nuclei also present in them, and they had portion of gray crescent present in them, and hence they could give rise to two normal uh, tadpoles. In the second case, he did again a bisectional division, but along another plane, and then he found unequal development. For example, if you make a constriction in such a manner that the nuclei or most of the gray crescent remains only at one side of the embryo, and the other side you make a constriction. So the side which has the nucleus as well as this gray crescent developed into a normal embryo, normal tadpole, whereas the side which didn't receive either the nucleus or the gray crescent completely formed an undifferentiated ventral part of tissue, which later on degenerated. So this again gives a double check that you need nucleus as well as this gray crescent region in order to form a complete entire set of embryo. Now what was the role of this gray crescent and how this blastopore lip concept came into a picture, we will study in the upcoming slide. This portion is now known as the embryonic induction, or the communication between the organizer or the inducer, what we call, and the recipient cell, the cell which are going to respond to it. How do the cells they tell each other that what they are becoming and what the other has to become? So one of the first experiment is Spemann-Mangold organizer experiment, the one which we studied in the previous slide. So this was done in the year 1924. The experiment stated that if you transplant the dorsal blastopore lip of one nuate embryo into the ventral section or another region of another nuate embryo, then he found that the host embryo, the one which received the transplanted section, now develops a secondary body axis, and as a result conjoined twins were produced. As a result, conjoined twins were produced. So the transplanted tissue didn't just grow back into anything. It was instructing the belly region, it was instructing the ventral tissue, the back tissue, in order to become a new neural tube and a spinal cord. And hence the embryo had conjointed larvae or the conjointed tadpoles. So this was termed as, this particular region, the dorsal blastopore lip, was termed as Spemann organizer, remembering his great remarkable contribution. He said that this particular region contains some of the very essential signals, a very essential proteins such as Gurdon and noggin, which helps to organize the entire body axis. Thus, this particular experiment received Nobel Prize in the year 1935 and was considered as one of the very basic experimentation in your embryonic induction principle, which states that cells can directly communicate with each other and they direct their neighboring cells and they can change the developmental fate. Those cells were known as the organizer or the dorsal blastopore lip. This is just a picture of that experimentation. So he took the dorsal blastopore lip from a donor embryo and he transplanted it into the ventral section, the region which used to form the belly of the host embryo. So what he saw, that two axis, body axis was getting formed. Because both were inducing the body axis formation, both were secreting noggin and Gurdon. So as a result, this made the formation of a conjoined tadpole having two body axes. So this region on his remembrance is known as Speamann's organizer. The next very important experiment is Nieuwkoop experiment, which gave rise to the Nieuwkoop center. The same blastopore lip, we will fine tune it as a Nieuwkoop center. So what experiment he did is, he took the vegetal cells and he transplanted them into the animal pole region. So we know that the cells which are present actually inducing the gastrulation thing is present at the vegetal pole side. So these vegetal cells, he took it and he placed them in the animal pole region, animal cap region. So he observed the formation of mesoderm induction occurred. He concluded that vegetal cells, they included the mesoderm formation, they induced the mesoderm formation. So if you take simply the animal cap and place it above a vegetal region of a cell, so you will find that normal complete axis was getting formed and normal larvae were getting formed because both were having normal gastrulation getting formed. In the second case, if you isolate this animal cell and you isolate the dorsal vegetal region, here also he found that the dorsal axis was getting formed, the dorsal blastopore lip was getting the normal gastrulation and everything was going up in a normal manner, and he said that Spemann organizer induced the formation of the body axis here also. And hence he called that there were some proteins present there, some information present there, and he called them as the Nieuwkoop center, which actually has the induction power. So it can, this vegetal region is known as the Nieuwkoop center and it was inducing some signals on the cells placed above them. And it was guiding them to form the mesoderm and then the ectoderm. The next important experiment is Briggs and King nuclear transfer experiment, considered as one of the main nuclear totipotent experimentation done in the year 1952. So in this experiment, he transferred the nuclei from a new blastula cell and he placed them into an enucleated frog egg cell. He found that the embryos still developed into successful individuals. The significance of this experiment was that it is considered as one of the first nuclei cloning experiment, and it tells the concept of nuclear totipotency. If you have a ovum having an animal pole and a vegetal pole, so he took a needle, he took the nucleus from a mature nucleus and he mature cell, and he placed that inside the ovum, inside the frog egg cell. So he found that after doing the transplantation, normal complete gastrula was taking place, normal complete blastula gastrulation formation was taking place, normal axis, body axis was formed, and a normal swimming tadpole was formed. The second case, what he did was just to see that how much is the potency of the nuclei. So suppose if you take the nuclei which you are transferring, this is an enucleated egg, and the nuclei which you are transferring, if it is at a early blastula stage, so those kind of nuclei were considered to have very high potency. They induced complete blastulation, complete gastrulation, and complete formation of an embryo. This success rate was as high as 40%. If you take a nuclei which is at a later stage, or any somatic cell nuclei which is at a later stage, so even though the formation of embryo could take place, but the uh, declining potency of the nucleus resulted in declining the success rate of the embryo, or success rate of the entire experimentation, and hence an abnormal development was seen. So these were the two subcases of this Briggs and King nuclear transfer, which is again considered as one of the very important steps because it is a first nuclear cloning experiment. Now, third set of experiments which we will be studying is about the positional information and gradation. So how do the cells then know where they are going to get placed in the embryo, so they can form the right structure at the right places? So those kind of experiment is known as the positional experimentation term. Here, the first experiment is Louis Wolpert French flag model experiment, which was done in the year 1969. So the model here is a theoretical model which explains that how the morphogen or cytoplasmic determinant, which are actually the signaling molecules, they decide or they dictate the cell fate. This was based upon the concentration of that particular morphogen or that cytoplasmic gradient present in that location. If a location is having high concentration of morphogen or cytoplasmic gradient, it is developing into, say, tissue A. If someone is having a moderate amount of morphogen, it is developing into tissue B. And if a region is having less amount of morphogen, it is developing into a tissue C. So high concentration of morphogen, the cells turned blue. They receive maximum amount of that concentration of that morphogen which is coming from the morphogenic source or from the inducer. So they will form a particular type of cell. The ones which are getting intermediate or in a concentration gradient manner, they will form the white cells, last region of blue to white to some initial part of the red, and the ones which received very less amount, very low concentration of the morphogen, develop into a complete C type of cell or a red type of cell, and hence this was known as a French flag model. This gives the cell a positional information that where they are present and how they need to respond to the concentration of the morphogen. So French American flag model. French flag also has red, blue and white color, and American flag also has red, blue and white color. So if you exchange some area between them, so how the things are going to get formed. Those kind of experiment is known as French flag model, developmental model, which is analogy or which tells you about the competence and genomic equivalence. What happens if you take these cells which are genetically programmed to be an American flag and you transplant them into a region which is destined to form a French flag? So both are having blue, white and red color, but the pattern is different. So if the cells are genetically fixed that what they want to form. So if you take a genetically programmed cell which has to form, say, the American part, suppose a cell which is destined to form the leg region, and you take it and place it into another region of the flag. Say you take and place it on the head region. So head part should form antenna and the leg part should form some clap-like region or nail kind of thing, the hind limb kind of thing. So what if you are exchanging the tissue? The concept here is that the American flag cells will read the positional information. That is, I am at a high concentration end. I have been taken from red region and I have been placed in a blue region. So they are now going to receive more signal from that morphogen. They will be at a higher concentration end, and hence they will respond differently. They will somewhat respond to this concentration gradient. But since they have their own genetic material also, so they will have their own genetic instruction. So somewhat they will form a mixed kind of structure. That means they will form like a star on a blue background instead of a solid blue strip. For example, this experiment is known as your French flag model, which we can see in a greater depth. If you see the French flag, here you have blue, white and red in a concentration gradient, and in an American flag also you have blue, white stars and the red region. So suppose you take a region from here, blue, and you place it in this region somewhere here between the blue and the white segment. So what happens now? This part is receiving more morphogen from this region, from the French region. So it is going to behave as per the organizer which is inducing it, but somehow it retains its own memory also, this star region. So it remembers that I am coming from an American flag. So it is going to form an intermediate kind of structure. For example, if you transplant the flank tissue from a frog embryo into the jaw region of a nuid embryo. So the region which is responsible for forming the flank is now placed in a region where jaw should be formed. So the nuid embryo will now misinterpret this information and it will make a mouth here. It will neither make a flank nor it will make a jaw. It will make a mouth structure, a mouth region here, because it will behave in a positional manner. Since you have placed it in a jaw region, it is going to form a mouth, but since it retains the memory of a flank tissue, so here also it is going to form a sucker kind of thing. So what happens? Although it is forming a mouth, since you have placed it in a jaw region, so it is forming a mouth, it is responding to the morphogen, but it retains its own memory. It knows that in my previous stage, I need to form the sucker part. So inside the mouth also, it is going to form the sucker region. Or suppose you have taken it from the say sucker, or say it can also form the balancer depending upon which embryo you have placed it. If you have placed it in a frog, so a frog has the sucker kind of thing, so it will form suckers. If you are placing it in a nude region, so it is going to form a balancer kind of thing. So this kind of signaling is universal and it is very species specific, that what structure needs to be formed where. So you have a nude embryo which has the, or which you have taken the jaw from the host, and you have the frog embryo which has this flank donor kind of thing. So once you place both of them, so it remembers that I am there in the mouth part, so somehow it is going to form the mouth region, but it also remembers that whether I need to form the balancer or sucker, and it will act accordingly. Clear? So this kind of experiment is known as French flag model. The next is John Gurdon nuclear transplantation, which is done in the year 1962. So in this experimentation, Gurdon destroyed the nucleus of a frog egg, and then he took the nucleus from a fully differentiated frog intestinal cell. So the nucleus from an egg or a completely undifferentiated cell which does not have, has not formed anything, it is an egg. So he enucleated that egg, he destroyed that nucleus, and then he transferred the nucleus from a fully differentiated adult frog intestinal cell, and he injected it into the empty egg cell. The resultant what he saw was that the egg, it developed into a normal healthy tadpole. That means that there is something present in the nucleus, what we call it as the genomic equivalence. So every somatic cell in the organism is having the exact information which is present in the egg cell, and hence the exact set of DNA helps the egg to develop into a complete normal healthy tadpole. Therefore, differentiation isn't about losing genes, instead it is about turning on and off some of the specific genes. So this concept means that Whether you take the cell from an intestine or you take it from a skin cell, they all have exactly the same complete set of DNA. And the moment you take them and place it in a nuclei, so the cells, they will just simply turn on, or the genes, sorry, they will simply just get turned on or off depending upon the stage at which they are developing, and hence a normal tadpole, healthy tadpole can be made. So this experiment is known as John Gurdon nuclear transplantation experiment. So I hope the session would have been interesting for you, and you must have learned something more about all the important experimentation which is taking place in developmental biology. Thank you.